Good afternoon, everyone. Thanks to all for joining another session of the NBS Distinguished Speaker Series. This series is part of our effort to promote cross-disciplinary discussions that can lead to new research and educational opportunities. The topic today is COVID-19 and its implications. I believe this is a very important topic for everybody at the moment. We are happy to know that this session has attracted many people from 14 countries and 19 industries. I'm Xin Chang from Nanyang Business School. I prefer people to call me Simba. I'm a professor of finance and associate dean for research at Nanyang Business School. I'm one of the two moderators today. Another moderator is my colleague at Nanyang Business School, Hong Ru. Hong is an assistant professor in the Division of Banking and Finance. He's one of our rising stars at NBS. He got his PhD from MIT, and he has published many papers in the top finance and management journals. Especially, I want to mention that in a very recent study, he and his co-authors examined the role of the SARS imprint in combating the COVID-19 pandemic. And they find that compared to countries that didn't experience SARS in 2003, countries with SARS experiences react in a more timely and proactive fashion to COVID-19 and achieve better outcomes. So the paper is very interesting and was accepted by Management Science for publication, which is one of the top management journals. At NBS, Hong is also the research director of the Center of Excellence, International Tradings and the Center for R&D Internalization Studies. Hong will give a concluding remarks at the end of this session as well. Okay, so it's now my great pleasure to introduce the speaker today, Professor Laurent Renia. Professor Renia is a professor of infectious diseases and the director of the program in respiratory and infectious diseases at Lee Kwang Chien School of Medicine at NTU. Before joining NTU, he was a senior fellow of A*STAR, the executive director of Singapore Immunology Network and the founder of the A*STAR ID Labs. He's a very prolific researcher. He has published more than 350 articles and book chapters. And his research has accumulated almost I think, 30,000 citations, according to Google Scholar. To me, is a very remarkable achievement. His scientific interests cover the immunology of infectious diseases to the development of new immunological assay and animal models that can be used to study communicable and non-communicable diseases, such as cancer. According to Professor Renia, his biggest career highlight is actually having two parasites named after him because he was among the first to identify them. And finally, I think I want to share with you that Professor Renia has played a very important role in helping Singapore combating COVID-19 since it started. He is a member of various committees, including National Center for Infectious Diseases Research Work Group and Therapeutics and Vaccine Selection Committee, the Vaccine Implementation Committee. And so we can infer that he was involved in a lot of high-level discussions on what vaccines to choose and how to use them in Singapore. So today we are very lucky to have such an expert speak about COVID-19. So his talk will last about 50 minutes, followed by the Q&A session moderated by me and Hong. I strongly encourage you to send in your questions via the Q&A tab. and so that Professor Renia can address them later. So without further ado, let's have Professor Laurent Renia.
Thank you, Prof Simba, for the introduction. When you mention like that my achievement, I feel I near retirement already. I will share screen. Okay, do you see my slide? I suppose so. And so I'm gonna select the pointer option. Okay, I think so we could start. And again, thank you for the business school to invite me and try to give you an update of where we are with COVID and where we are going and what are the implications beyond looking at the disease. So I have a disclaimer to make because I've been involved, as Prof Simba mentioned, in many working committee on COVID research, but also on vaccine selection, on vaccine implementation. So you might see sometimes that maybe my views are a bit biased, but I was in the decision in some of these committees. So I think it's important to have a disclaimer. And I've also been in a committee to give opinion on the vaccine from Johnson & Johnson. So my views are, for this talk, are only my views, and they don't reflect the position of the different committee and the different bodies I'm being part. So I think it's evident from all of us that really this pandemic for the last 2 years are changed our life, our economies, the way we interact with others, our practices, just to mention mask, for example. And for many, I tend to say they're common senses. Even for people who are a bit knowledgeable in the field, sometimes I read things which I don't believe they would say or even think. But more deeply, this has revealed the weaknesses in various health systems. And there is a whole economics of the health system, which I think has to be completely thought again and maybe refunded. The weakness of our scientific education, if you look at the social network, you could read or listen to things which you, in a normal situation, you would say, my God, what people are thinking that. And I said, you could, using social network to see the good, you could get good and reliable information, but also the bad and ugly sides on crazy discussion or even violent discussions. But at the end of the day, the COVID-19 pandemic just highlight that we need to have multidisciplinary approach, not only medical or biological, because evidently we need to understand the virus, but we need to understand the effect of a pandemic in multiple aspects of in the society. It could be economical, but it could be societal and so on. So where are we now? If you look at the situation in the world, and you could see that we are over now 350 million confirmed cases, more than 5.6 million cases, and you have on the left side here the two plots, and we see that the curve is not plateauing yet when you look in total number in an arithmetic way. But if you look in a logarithmic way, we see that progressively we are starting to reach the plateau, which is in a way a good sign that the epidemic will at least stabilize. I don't know if it will completely stop and decrease, but at least we are nearing stabilization. And when we look in Singapore, where we had a lot of cases, around now 310,000 confirmed cases, but we were able to really maintain a very low level of deaths below 900, which I think is maybe people don't realize is a big achievement when you compare to other countries. And again, if you look at different presentation, even if we had a blimp at the end of last year, mainly due to the Delta variant spreading into the community, we still maintain a lower level of death. So again, even if we don't have a bad variant, we're still in a good situation. Overall, I would say that in Singapore, the pandemic was well managed. So where were we in 2019 when the COVID situation started? We knew nothing about these viruses, very little about the biology, nothing about the etiology. Etiology is the science who tells you what an infection will give you as symptomatic signs, as number of deaths, which age population, epidemiology, how this virus will disseminate in a population, what type of subgroups it will eat. So we knew nothing about that. We had no really rapid detection test. One test was the PCR test that I'm sure you are all familiar with. And luckily, it was developed very fast. We had no what is called point of care test that I'm sure most of you now are familiar with, which are the ART, automatic, personal test that you could buy in a pharmacy or get, and you could test alone. So we didn't have all these, which really helped to track the number of cases. We had no validated treatment. A lot of people proposed various treatment and so on, but none of these were validated, and it lasted at least for the complete year of 2020. And prevention, we didn't know exactly what public health measure to take. And I will give you some example. Do we need to have social distancing, 1 meter, 2 meters? Or if you are too close, will you contaminate more people? What is the mode of transmission of the virus? Through contact, through air. And we didn't have vaccines. So coming back to the Singapore situation, really until March 2019, remember that the first case was in the beginning of January 2019. I think it was in January 8, if my memory is good. We were okay. The government limited the number of people coming in. There were a lot of control at the border and so on. So it was well managed, but you could not block completely the borders, because Singapore is an open society, relies on trade, so it has to be open. And if it's open, you know, the virus, they will find holes. So it did that. And in fact, even if in the community it was okay, but people forget that there were the dorms. And why the dorms at the beginning was a big problem, because in the dorms you have a high density of people. And pathogens like viruses, they like high density of people because it favorizes transmission between people. So again, to extend, when you want to deal with viruses like COVID, you need global and integrated approaches. And one, I would say, outcome of this infection is the government is really rethinking the way the dorms are going to be organized and how many people you will put in the dorms and so on. Because there was really an Achilles heel in the way we were managing the pandemic. It was more an outbreak at the beginning. And for that, you really need to go beyond biology or beyond medical knowledge. You need to have public health with social and economics knowledge. And when I mean social is also behavioral knowledge. How do people interact in one environment, and how maybe by changing their behavior, you could really limit the spread of a virus or another pathogen. And I think you all remember this painful moment where we had the circuit breaker, because if I come back to the dorm, the number were crazy. It went up from 5-10 cases per day to thousands of cases. So the government needed to really make a break, and that's why we had the circuit breaker, which worked. You could see that the numbers went down and to a more manageable situation. But for all of us, it lasted nearly more than 2 months, and it was not very easy to go for shopping or even exercise. And then he brings us back to something that people tend to forget, because when there is no outbreak, no pandemic, life goes as usual. But for Singapore, infectious disease has to be a national strategic imperative. As I said, for the dorms, if you look at the population density in Singapore, it's one of the highest of the world, which means that for any easily transmissible viruses, you will have outbreak. Another aspect is that because it is an open society relying on trade, Singapore has always been a global transportation hub with highly porous air. You know, at the best, Changi will have 50 million passengers going through Changi every day. But we have also land and sea borders. And beside the airport, the port in Singapore is very important. And so if you go back to the recent past, and I will start with SARS-CoV-3 in 2003, SARS-CoV in 2003, you could see that we have been submitted, or we have been suffering for a string of infectious disease, either viral, either bacterial, either parasitic. You know, after SARS in 2003, we have a peak of H1N1, but this didn't have the size of COVID that what we are experiencing now. But still, we had outbreak could go from 50 people to thousands of people, but they were constrained in time and they went away. But we had chikungunya in 2007. These are mosquito-borne. Malaria outbreak in 2008. Another flu virus, H1N1 in 2019. Scares of, luckily didn't reach Singapore, of Ebola in 2010. And then Zika, if you remember, in 2016, one case of important monkeypox, GBS bacteria in the food court with 5 or 6 people who needed to be amputated to stop the spread of the bacteria in their body. And in 2020, SARS-CoV-2. So you could see, because of our geographical and particular status, Singapore is really a hotbed for emerging infectious disease. And so that really need now to take into the mind of many people and think how we could really change the way we think, change the way we envisage infectious disease, and change the way we are going to deal with them, because this is not, the list is not going to end in 2022 when COVID will progressively disappear. There will be others. However, and I think it's good that Prof Simba mentioned that Prof Ong wrote an article on SARS, because SARS was a turning point already for Singapore. SARS hit Singapore very badly. It was bad because more than 30 people died. It was mainly doctors and healthcare workers, because they were the frontline people. But these really raised an alarm at the government level, in the Ministry of Health, that we need to be prepared. We need to be prepared. We don't know when the next one will come, but we need to be prepared. And you know, the next one was in 2019, the COVID-19. So, but what SARS did was that it led to the creation of the National Center for Infectious Disease, an institute which is really dedicated to deal with patients arriving with known disease, but also with unknown disease. And this center is really the progeny, the child of Professor Yi-Sin Leo, who was the head of the CDC in Tan Tock Seng Hospital, and really fought very well to get the funding and to make it happen. So if you pass near the NCID, please remember that it's just in front of TTSH. And so it's organized that way, and there are multiple subgroups. I will not go into details, but some looking at bacterial infection, HIV, which is still endemic, dengue, which is still an endemic disease in Singapore, but also different structure which could be really mobilized when there is something like COVID happening. And another aspect was that in 2017, again, with these members that you see, people from NCID, Prof Leo, Prof David Lai, or Prof Mark Chen, the then director of Biomedical Research Council of A-STAR, Professor Bensit, and Dr. Vernon Lee in MOH, we were thinking of, can we have a program to really be ready for any outbreak and anticipate? So we wrote this project called Orbits. It took us a lot of discussion back and forth with the ministry, and we were on the verge to be funded when COVID-19 arrived. So this project was put aside, and because we had to respond to the pandemic. But you see, we were already preparing our mind for something like that, and how we could mobilize people and respond. And just to show you, there was a sketch that we drawn at that time on what do we do to have an outbreak response? How do we organize with not only in Singapore, but also at the level at the region, the ASEAN region, and also beyond, to get the expertise that we don't have in Singapore, to get the tools that we don't have, and to respond very, very fast, and how we could organize cohorts, how do we deal with the new pathogen, and so on. So that being said, because of this group, when COVID hit us in January, we were ready. NCID started the 4th of January, the NCID COVID research group that we call the Protag Project, which the idea of every cases coming in Singapore will be sent to NCID. And we brought different people from all the university, Duke, NUH, and from A-STAR. And this group will meet at the beginning even once or twice a week and organize the flow of samples, discuss the clinical cases and so on, to really start to build an adequate response to COVID. And we still have, now it's bi-weekly meetings where we review the situation in Singapore, but also the progress in science in Singapore and in the world. So we could tailor our response to the Singapore needs. So now if I look at where we are now in terms of the need, those we have partially met, and those where we still need to do better. We know a lot about the biology. We know how the virus is transmitted. We know the epidemiology, what are for example, the group at risk. We know the elderly, the people with comorbidity, diabetes, obesity, and so on. We have a lot of rapid detection tests based on RT-PCR. We have faster and a large variety of laboratory molecular tests, but also point-of-care tests, which are specific, sensitive, and cheaper than the one we were developed at the beginning. Now we have treatments. There are small molecules which are coming from Pfizer, for example, and we have therapeutic monoclonal antibody, and I will go later to explain what they are. And we had what I call the first generation of vaccine, either based on mRNA or inactivated. So we have made progress in 2 years, and this progress have really made a difference. You are not doctors, so I will not go too long into that, but if maybe you know now, but just as a recap, that the symptoms when you are SARS-CoV-2 are cough, shortness of breath, and a running nose and sore throat. So if you have that and if you suspect you have also fever, please go and test. But you could have other symptoms like nausea, and you could lose your sense of smell or sense of taste, which is called hypogeusia or hyposmia. And you could have a wide range of other symptoms like fever, muscle pain, fatigue, headaches, confusion. So we do the diagnostic by PCR or by the point of care test. We do biological test, but it's more for the clinician to follow. And we could confirm using check-stray that your lung has a problem or not. So we know now a bit more about the causes for all of these symptoms, and that allows the clinician to provide the better treatment. That's why also something important to say, clinicians are losing less and less patients, even when they go into ICU. So I will give you a short overview of the immune response. And I think it's important for you to understand. I'm not going to give you a lecture of what is immunity and so on, but rather a glimpse of what is the immune response, how that it is induced during the infection. And when you will look at the newspaper and they will talk about antibody and T cells, you will have an understanding of what is happening. So first, there was one study, for example, that we did at the beginning of the infection when we knew nothing. You could take blood of people arriving at the hospital and you could measure protein, what we call mediators, immune mediators in their blood, using a particular technique. I will not go into details. And if you look at the graph here, the dotted line here are what we call the normal people. So we had blood samples from healthy individuals not infected with any other disease. And it may be evident for you when you look at these graphs that in all, and the first cohort that we have was we had 81 patients, that all these markers were above the dotted lines, meaning that these people already had a particular response. And when you look at what these molecules are and what they represent, it's what we call an inflammatory profile. Inflammation, for those who are not familiar, is what gives you fever, or what gives you the rashes, for example, when you are bitten by mosquitoes. And so first indication that we have, these people have a strong inflammation. And when we look a bit deeper by classifying the different people depending on their severity, the healthy control on the left, those who were with fever, myalgia, but don't have pneumonia, the difficulty with breathing, those who have pneumonia, when you do a chest X-ray, you could see on the image that their lung has a problem, but they didn't need to be intubated. And those who were intubated, you could see visually that they have really a different profile in terms of these markers. So for us, they really tell us that the people who are really sick, this group here, have a very strong inflammatory profile. And that was described in literature, but you have also maybe seen these in the newspaper, what is called the cytokine storm, when your body cannot control this type of response. And instead of helping you to block the virus to multiply in your body, this response attacked your system. And one application, you could say, okay, it's nice to see that, but how can you use that? But one application that there are anti-inflammatory drugs that you could take. One is Panadol, especially when you are in the middle here with no pneumonia. You could take Panadol, and that will reduce this inflammatory profile. But when you are in the red part on the right, some of the drugs that could be used are corticosteroid. And one drug was shown to be very effective was dexamethasone. And now in people with ICU, they are treated with dexamethasone because we have this knowledge now of this group. So again, just to give you an overview, and if you have to remember something, is just that the disease process is very complex. So if you look on the left, this is your normal lung. You have the hair coming here, and it's going from the lung alveoli to your capillaries, and that's why bring you oxygen into your lung and release of CO2 into the alveoli, and that's what you exhale. But when you have infection, when you have a virus coming into this environment here, you have a lot of cells, a lot of molecules going there, and this creates this mess that you see here, a big reaction. And we know that this creates a cytokine storm, which is responsible for the disease. But we also realize that there were one type of mediator which was missing, which is called interferon. And this molecule is like your bodyguard at the door. If a virus want to enter in a cell, the cells will produce this molecule and prevent. But for a strange way, this virus is able to make your body not make this molecule. So it's like the virus removed the bodyguard, so the door is open and it could enter more easily into your body. So these give us also ideas of maybe if we add this type of molecule as a treatment, we will protect. And there have been clinical trials seeing that if you treat people with interferon, they could be better when they are infected. So the previous part is what we call innate immunity. It's what happened the first days during the infection. But there is a second part which is very important, especially when you want to develop vaccine, but also for you when you have been infected and your body responds correctly and help you to get rid of the virus. And what is called adaptive immunity. It's adaptive because it takes more time to develop than the first type of response like I was showing you before. And again, if you have to remember only a few things, is that you need four things important. You need antibodies, and these antibodies are made by these cells, which are called B cells. It's like the B cell is like the factory making the antibodies. But you need another type of cells called T cells, and they come in two flavors, either CD4 or either CD8. And these cells are important for one thing. The CD4 are the workers which help the B cell factory to make the antibody, and the CD8 are the killer. They recognize the infected cells and destroy them. So preventing the virus to multiply and to disseminate into the different organs. And so to make something short and simple, when you are infected here with the virus, and the virus enter your cells by recognizing a molecule on the surface, see it as the virus as the key, and on the cell surface there is the lock. And so the virus use the right key to go into the lock and to enter into your cell. It's like enter into your apartment. And then it will hijack the interior of the cells and make the people, in this case, the cell machinery, work for it and create more and more viruses. And that's what viruses are very good at. When they are outside, they are useless, but when they go inside, they hijack the cells and make them work for themselves to replicate and disseminate. But you could fight back, and that's what your body is doing. It's making these T cells that I say, which are going to kill the infected cells, to destroy the infected cells, or they're making these antibodies through the B cell factory, which are going to coat the virus and inactivate it and prevent it to go inside the cell. So you need the antibody, you need the T cell. And so because we learned very fast that the antibodies were important, some researchers went into antibody-based therapy. And there is one therapy which has been used extensively in the past, where you take blood and you separate the cells from the plasma. The plasma is what you have in your blood, and is the liquid form. And then the plasma contains the antibody. And in many other diseases, you could take this plasma and inject back in people who are infected. And for diseases like hepatitis A, other like tetanus, you could prevent the infection because the antibody will neutralize the pathogen. You could be a bit more clean because your plasma contains the antibody, but also many things. But you could make preparation a bit more clear. It's what is called intravenous immunoglobulin. Immunoglobulin is another name for antibodies. And you could purify these antibodies. You could concentrate them and again, inject them. For COVID, I must say that the results have been for both convalescent plasma and intravenous immunoglobulin. The results are contradictory. Some studies report that it worked. Some studies report that it was not very efficacious. So up to date, we don't know what to conclude. Again, using the same approach, you could make animals produce these antibodies. You inject them with the virus. And most of these animals, like llama, alpaca, camelid, or even horses, don't develop the disease, but they will respond to it by making antibodies, and you could purify these antibodies. Again, to give you an idea that it's not new technologies to treat tetanus when at that time where we did not, in the 19th century, where we didn't have either viruses, either vaccines or either antibiotics, because tetanus is a bacteria. So that's what people were doing. They were infecting horses, taking the serum from horses and treating people. And this was working, but they had a lot of side effects. So that's why we evolved with something more clear. So there are companies, for example, using antibodies from pigs which have been humanized. I will not go into details how they humanize the pigs, but they make antibodies which are closer to what we are making as humans, and they want to treat with these antibodies people. They have data in an in vitro system, but there is no data on efficacy for the moment. But it's one of the possibilities of the future. And the one which works, and now this has been validated and commercialized, is human monoclonal antibodies. Companies have purified these B-cell factory that I was talking about from humans, isolated by genetics manipulation, genomics manipulation, the genes, express them, and now they could produce in large quantity very pure monoclonal antibody, which are specific for the virus. And so if you look in the blue here, that's where there's different monoclonal antibodies. You have a list here. I will not go into details. Many companies, AstraZeneca, Brie from the US, from China, from Korea have made. And they will treat the people at different level of their infection. But what is clear is that if somebody who is not vaccinated or doesn't respond to vaccine and is diagnosed, you could treat them after exposition to the virus or even at the early stage when they are diagnosed, and the success of this treatment is very high. You could even use this antibody as what is called pre-exposure. Let's say you are in a group, for example, a dorm, and you know that you have a big outbreak there. You could treat other people who have not been infected but are in contact. And then even there, you add to these people another layer of defense, and they will be protected against infection. So my part will be now on the vaccination. Just to recap for people who are not used for what is vaccination, is a simple concept. And that was put out by Edward Jenner, a British clinician, that you take something which mimic your infectious target. It could be an inactivated, attenuated, a part of the virus, a part of the pathogen you inject. You know, by injecting it in a naive individual, you're going to induce an immune response. And this immune response gives you memory and protection. Why is it better than to be infected? Because when you are infected, the adaptive immune response takes 7 days to 10 days to develop. But if you have memory because your body have seen the virus or something which is close to it, you're going to respond in 24 to 48 hours. So you shorten the time of response and you block the virus at the beginning. So what you want for an ideal vaccine, that it is long-lasting, that it recognizes in the case of COVID all the viral strains, that what we call immunogenic is that everybody who is going to be injected respond, that is safe. You don't want to have a molecule which is giving a lot of side effects that will break what we call the risk-benefit. You want something which gives you more benefits than risk. You want something to be easily injectable, not intravenously, not inside your body. You want something in the muscle, in the skin, or even to take orally, easy manufacture and low price, because you want vaccine equity and be able to distribute to low-income country, because you cannot treat a disease locally. It has to be worldwide. You want to get rid of a pandemic. And the best is to be stable at room temperature. And if you remember the discussion with the Pfizer or the Moderna vaccine, they had to be refrigerated at -80. And that is a logistical problem. Something which was incredible during COVID, usually if you look at the development of a vaccine for any disease, it will take 5 to 10 years at best to go. I'm not even talking even licensing or commercialization of the vaccine, but just deployment, what we call mass vaccination. And for COVID, this was shortened to 6 to 9 months. This is unprecedented. But the drawback of these is that because the mRNA vaccine were experimental, even if they have been around, the concept of mRNA vaccine have been around for 10 years, and they have been preclinical data, even a small phase 1 clinical trials in human, because it was so fast to be mass deployed and we don't have enough time to evaluate long-term side effects, that created vaccine hesitancy. And still up to date now, when we know after 2 years that billions of people have been vaccinated, vaccine hesitancy is still a big issue, which is understandable because the time to develop this vaccine was very short. So there is a list there of vaccines, but again, I will not go into the detail and listing all the different characteristics, but you have to remember that you have 3 categories. mRNA vaccines, which mRNA is the piece of the viruses which contain the information to make the protein, which is the molecule which is the one will induce an immune response. Adenoviruses are other viruses that have been modified to express the protein of interest. In this case, is the spike protein, the protein which covers the virus, and was shown to be a good target for antibodies and T cells. And these have been the mRNA from Pfizer and Moderna, and adenovirus has been from AstraZeneca and Johnson & Johnson. We have the inactivated viruses, mainly the Chinese Chinese vaccines from Sinovac, Sinopharm, but also there are companies in Europe like Valneva, which has made these type of viruses. The difference that all the viral protein will be expressed because you use the whole viruses, and it gives a larger immune response, but seems to be at the end of the day a bit less efficient than the mRNA or adenovirus. And the new kids on the block coming, like Novavax, which has been accepted in Singapore and should be deployed at the end of the month, is a protein-based vaccine. This is really a standard way of making vaccines. For people maybe in the audience who have been vaccinated with hepatitis B, hepatitis B vaccine is a protein-based vaccine. So we have more knowledge about the side effects, about the way, the stability, and so on. So if people are still hesitant to go for mRNA and adenovirus or even inactivated, I will recommend you to go for protein-based vaccine. Get vaccinated. So what does the vaccination do? You inject in the body these antigen, as I mentioned, different forms. And you're going to create memory B cell, the factory, to make the antibodies, and the T cells, which are needed to kill the cells. And you could see here on the graph below that the vaccination will induce a response. You boost first one, you induce a better response. And that response here, that this peak is necessary to control the virus. And you know, this response will wane over time. So we were a bit disappointed that it went faster for the mRNA and the inactivated, because in some other vaccines, it could last for up to 10 years. And then that's why you will need boosters, is to bring your response to that level where you have protection. And it's the same for T cells. You could see here, if you have a primary response, which means you're not vaccinated, your T cells will come later after the infection. But if you are vaccinated, you make this big T cell response, which control the peak of the virus and help you to eliminate. So really the vaccination induces these antibodies, which are necessary to block the virus, prevent it to go inside your cells, and induce the T cells, which will kill the viruses which are succeeded to infect your T cells. So what are burning questions now for the vaccinations? I'm sorry if the graph here is a bit complex, but it's just to give you an overview. You don't have to remember everything. There are positive drive for the vaccine effectiveness. They have negative drives, and we are unknowns. So we know, for example, in red, that the vaccine efficacy is limited by the fact that you are older. We have done a study that we see that if people are over 60 years old, an age I will reach soon, so I'm going to go into this category, not too long and not too long. And then you respond a bit less well than if you are a younger person. If you have previous infection, the vaccine will boost your response better. So that's a positive effect. You are not equal, and some people will respond very well to the vaccine and others will respond much less well. And it's because our genetic background is different. And some also other people have different underlying health conditions. If you have cancer and you are treated, or if you have autoimmune disease and you are treated with immunosuppressive drugs, you will respond less well. There are other factors like demographics. Again, if you have a large outbreak, the vaccine will be less efficient because you will be exposed to more and more viruses. And so there are, just to give you different aspects. And the last one, which is really the problem, is the viral variants. This virus is not something, even if outside is inert, but when it goes into your cells and multiply, this virus will change. While the virus which enter your cell and the virus which exit your cell is slightly different. And these variants which are created by every time the virus multiply, and some variants will be bad and die. They will have no capacity to infect further, but some will be with new traits, better transmissibility and so on. So I will give you a bit of oversights of the variants, because I think it's important, again, because we are in Omicron wave, which was the last variant we were confronted to, but we may have other variants in the future. We all hope that, and myself first, that this will be the end of these continuous flux of variants, because we will have enough immunity in the population and we'll be able progressively to decrease the number of people infected and decrease the number of people being able to transmit the virus to this population. So first of all, when these variants occur, here on the right, you have the schematic of what is the virus. You have this crown with the virus, the S protein on the top. I said this S protein is the target for immunization. And inside you have this RNA molecule which contain the information for the virus to create, when it enters the cells, more viruses. And if you look at the surface of the S protein, that's how it looks like. It's 3 molecules packed together on the surface. And that's contain the information on the top here to recognize I will say here is the key, and that the key recognize the lock, which is the ACE molecule that is on your cells. But first thing that we look at when there is new variants and we sequence very fast this structure is first, is there variation in this region here that we use to amplify, to identify the variants by RT-PCR. And some of these variants have mutations there, and the RT-PCR was not working, meaning that we were missing the variants, so we were having the wrong diagnosis that people were not infected when they were infected. So that could be fixed rapidly when you know, and you do that. And so I'm going to have to speed up a bit. So when we could track the virus evolution, you could see by sequencing, and you could also track by comparing the data over different countries and see that the virus is really spreading in the world and say that these variants come from where and is going from where. And if you look at this database, you could see now here that the Omicron is taking over. Here you have the different waves of variants taking over the world when you look only at the sequences. And what matters is because in a natural host, these variants will make new viruses, and the vaccine, in fact, is like a gel, and you want the virus not to go in the population. And you want also that the new virus, doesn't mutate, that you will have an increase in pathogenesis, an increase of transmission. That's what we have seen with Omicron. So tracking the variants is important. So back to normality for us will mean that we will look for correlates of protection. We know antibodies are important, but we don't know what is the level that you need to have, which is important. So we really are tracking these and are doing more studies to determine that. Because if we have a simple assay telling us that, oh, we need that level of antibodies, we could design a type of ART which will say, okay, these people are protected or will be protected, these people will not be protected, so maybe these people need to be boosted. So we will not be able to boost all the population, but to really target the boosting to the people who will need it. So a duration of the immune response, for the moment, the data says that it's 3 to 6 months. So that's why we need boosters. And that's why it's considered now that what we call the 2-dose regimen at the beginning of the vaccine campaign was that 2 doses were needed. Now we're seeing that 3 doses are needed to at least give you a long-term protection. We don't know yet if after the third injection, and studies are ongoing, we will maintain a high level of antibody response and T-cell response, and people will be protected. So something to watch in the future. We know there are age difference, and I told you that over 60, the antibody went faster, or the people have difficulty to really mount a good response. So booster injection will be needed. But not only, there are research now being done to say, okay, we have good vaccine, but can we have better vaccine, which will induce a better response, a long-term immunity, and touch all the populations? Again, something to watch. And the last point, herd immunity. This is concept that if you reach an immunity in more than 75% of population by either people have been infected and they have recovered, or either people are vaccinated, progressively the virus will wind down because it will have no new host, naive new host to infect. So far, there are no really sign of this, especially with Omicron, when we have a large coverage of vaccination. And you could see that the virus, because it mutated, was able to infect people who had even 3 injections. And I will go to something which has been with the vaccine, since we know the vaccine, that the anti-vax movement, And you always have people, you will always have people who will doubt of the vaccine. And this is not new. That's on the left, you have a picture from the 30s when they were vaccine against polio, against smallpox, and you had people really fighting in Brazil against the mandatory vaccination. And you even had at the beginning of the century an anti-vaccination society in the US. Really, we were fighting very strongly against a vaccine mandate. So nothing new here. And the only things you can do against that is convince people about the benefits of the vaccine. Vaccine is not perfect. As I said, you have benefits and risk, and you have to really make the balance. And that's why scientific education can give you an educated guess on what you should do and what you should not do. So the 3 minutes I will go for the post-pandemic world, and I think that's all where we want to reach. What do we know for human coronavirus? SARS-CoV-1 disappeared. It was very transmissible and it was very controlled because people, when they had signs, had to be isolated. And then this break very fast the dissemination and helped to get rid of it. We have viruses of the same family which are circulating. That's what we call endemic, but they give disease in a small population, NL63 or C43 and so on. They don't induce lax immunity, so there are cycle of reinfection, but small outbreak. And these tell us that we need to do targeted boosting. And what do we know? What we don't know is we may have future waves of variants, and maybe we will need to develop either variant-specific vaccines or pan-corona vaccine. Variant-specific vaccines exist already for flu, that you need to have a flu boost with a different vaccine, flu vaccine. And I think that vaccine strategy much focused on severe disease because that's the people who are really at risk, that they need to be targeted. And vaccine equity is also very important. If you think that vaccinated only rich country will save us for the next variants is a mistake. So what are the pandemic possibilities? That's the peak and valleys that we are in. Maybe we hope to get into progressively either the peaks and decrease or slow burns if the virus become endemic. So what did we learn and how we prepare for the future? If you look at this map here, you could see that there are emerging and re-emerging disease all over the world. And even if we had this map even in 2017, this map was done in 2017, we didn't see SARS-CoV-2 coming. We know, if you look in the middle here, we know that the intertropical region is the region where most likely these emerging diseases will come from. So they have to be watched. They need to be surveillance. We know also from history and with the extent of traveling, fast traveling with plane, that you could see that viruses and diseases could disseminate, that the one in the middle where when traveling was limited, you could see it was regional. But now, if you look here with SARS, how in less than a month, this disseminated around the world very fast. We know that most of the disease emerging comes from animals. You could see here the different countries where they have emerged, could be from domestic animals. So we need surveillance, but not only domestic, but wild, could be camel, could be pangolin, could be duck. But you need to have surveillance and not limited to human, but also to animal. And it's called the One Health concept. You need to really track this disease. And there are multiple reasons for emergence: market, wildlife farming, wildlife hunting, and also, and that's something which is important to take into account, as the moment you're going to change your environment by cropland change, population change, deforestation, you're going to have an increase in the probability of emergence of these, what we call zoonosis disease, which come from from animals and go into human. And so just again, a recap, country, we have mosquitoes, and some mosquitoes we were adapted in the forest. They could adapt now in the city. And one good example for you who live in Singapore is Aedes mosquitoes, which were countryside mosquito and have moved to the city and are well adapted. And go. So you need to have research needs involving climate, material science, geography, economics, big data analytics, and to really approach the problem by different angles. And if we have serious, we could fight back. You could see that at the beginning of the century's data from the US, the mortality was more than 800 by 100,000 people, very high death rate, and we had different peak of pandemic influenza, but the introduction of sanitation, antibiotics, and vaccine really decreased the number of infectious disease over the years. And that's why we have to do. And Singapore is planning for the future. We have a PREPARE program, which derived from the Orbits program that I showed you, where really we will look at enhancement and surveillance and early detection capability, look at diseases and prepare the technologies for therapeutics and vaccine and have better diagnostics. And we cannot do that if we are working only in Singapore. We need to be integrated in a larger context regionally. And that's what is being planned. So next time we may be ready to fight against a new disease threat. And I will terminate here. And if you are vaccinated, well done. And if you don't, please go and get vaccinated. And don't forget, there is a Novavax coming. So if you have still doubt and you want something more traditional, the Novavax may be the good one for you. And thank you for your attention. And I will take any question. And sorry, I've been a bit long by 2 minutes.
Okay, thank you very much, Professor Renia, for such an insightful and informative presentation. So I personally learned a lot from it, as many audience in this webinar, I guess. So let's start the Q&A session, and please allow me to ask the first question. So as you mentioned in the presentation, looking back in history, the pandemics happen once or twice in a century, the global pandemics, and the epidemics happen almost every decade, several times. So besides the government actions like the closed surveillance system you mentioned in the end of the presentation, how can individuals like us from a social perspective to do better to prepare for the next outbreak or even prevent the next epidemic or pandemic? Thank you very much.
You see, thank you for the question. This has multiple layers to answer, because prepare is, it's going to be at different level. The first one, for example, when you are in the middle of pandemic, social distanciation is a personal thing that you could do. Wear your mask, be responsible. That's something you could do to reduce. Good sanitation, you don't throw your waste everywhere. You have simple thing to do, but I would say that simple thing to do on your daily life to prevent the disease to disseminate. At the personal level, if you want to be more invested, I finished, I'm sorry, I was a bit rapid at the end, but modifying the environment is one of the major cause of emerging of zoonosis. So maybe, when there is development and the development is, okay, it's necessary for growth and so on, but, it's like for vaccination, you will have to really think is it going to, the balance between benefits and risk is going to have to be taken into consideration. I could give you one simple example, for example, of you have to take, to think of environment. People were building these big tower and so on with the refrigeration system and so on. And then they realized with the refrigeration system, one type of bacteria, which is called Legionella, would grow very well in this system. And you had outbreaks of Legionella. Nobody could understand at the beginning where it come from. And then they track and they found it was the regeneration system. So what they did, they installed filters and so on. So for every disease, you need to have the knowledge of the biological knowledge, but there will be implication in terms of material science, in terms of for economics, you have to take the cost of having extra filters and so on into the system. So you need to approach that from multiple angles.
Thank you very much.
Good. Yeah, thank you, Lauren, for the great presentation. I was quite amazed by how you condensed so much information into a 15 minutes presentation. Especially I like the part in one of the slide where you mentioned that, in this fight against COVID-19, we need to take this multi-disciplinary approach. In one of your final slides, I saw some discussions about how scientists in different fields should actually work together. Maybe you want to elaborate a little bit more on that. And then especially, I hope to know, like, for example, what's your view on what are the potential areas for the collaborations between, scientists on infectious diseases with people, business people or business researchers in particular?
There is a field which is growing progressively is medical economics. For example, the biotechs, big pharma, they always approach the problem of diseases in revenues. I'm going to create a product, it's going to bring that amount of money back to me and so on, and I will do R&D and make new products and so on for different diseases. But there is a thing which is for government and which is very important is if I do certain measures or I implement certain measures, how much money I am going to save. For example, if I go to the hospital and I have an IRT test and I triage people, those who were sick and not sick, and I only select those who goes to the hospital who are sick. First, I'm not going to have the waiting list of people or the waiting room waiting 20, 30, 40 minutes. How many nurses are going to be needed to take care of these people? How many staff clerk are going to be needed? What is the time of the doctors being taken to take care of these people? So how much money do I save? So if I have other intervention, you were talking about diseases, but it could be diabetes. If I change my attitude, if I eat better, how much money I gonna save? So medical economics, not so much how much money we're gonna make, but how much money we're gonna save. It's really something which is needed for the future because we cannot grow a medical system. If we had one NCID was one response, but imagine we have a disease which is twice the size of COVID, which is already big, but twice the size, and NCID is overwhelmed. Our other hospital is overwhelmed. How can we manage that? What is the response? Is to create another NCID? No, there is one moment where the health system cannot grow further. So the approach will be that for infectious disease and other, what are the measures that we could implement? And that could be, as I said, you could have medical, biological measures, but you could have other measures. And it could be management measures that will reduce the cost and make a pandemic manageable.
Yeah, that's a good idea. Essentially, I think you're saying that it would be nice that we can quantify the economic impact of measures against this infectious diseases.
It's something which strikes me during these pandemics, and which is very different from the other one. The real readout for any government in the world, maybe not in Africa because they don't have the capacity, but was how many ICU beds do we have? And how many of these ICU beds are occupied? Mm. And all the measures that we have were dictated by this number.
Wow, okay. Yeah. Well, I didn't really realize the importance of this particular measure. Well, after you tell me, of course. But imagine we can quantify the economic impact of different measures. I guess then the policymakers can make a trade-off between like different alternative solutions, I guess. Well, then we have a follow-up question about, for example, many industries can contribute in different ways to this fight against the pandemic. What's your view about the finance industry? Because I'm a finance person, Hong is also a finance person. I know you are not a finance person, but maybe you want to share your view first and see whether Hong and myself can have anything to add.
You know, I saw that when I've been a biologist all my life, but when this started, I have friends in the French community in Singapore, but elsewhere, who are either in the insurance field or bankers and so on. And I've been very surprised that now they ask me to give talks because they want to see when the pandemic is going to stop and what type of scenario they could envisage. Because I think for a lot of them, they are thinking of investment and they want to know where to invest and they want to have some kind of feeling, are we going to the end? Is it going to continue? You know, it's like they are really integrated the risk because like in finance, you always measure the risk also. Yeah. And they have really integrated infectious disease now as a risk. And I think maybe not all of them, but for many of them, when they're going to start to build financial projects and so on, they will include infectious disease risk, especially if they go to some regions where maybe the next wave of pandemic could come there.
That's a great idea. I think we need some people to estimate the exposure to this pandemic risk. I guess, to my knowledge, nobody has done that yet. I think to add to your answer about what actually finance industry can do, I guess in recent studies, we focus on how fintech can contribute to the fight. So basically, essentially, we find that, for example, fintech can help distribute the digital consumption vouchers to consumers and identify consumption patterns. And more importantly, fintech can actually improve the people's access to financing. This will help the resilience. And then there also research emerging talking about the importance of social bonds. Social bonds, the bond issue to address social issues, especially during the pandemic. And then, I guess a lot of bond issue to address is especially inequality, the inclusion and diversity issues. I think these are, I guess, the things that finance people can do, what finance industry can contribute. So Hong, you can.
Again, I've been surprised is there is something where the finance environment is well aware of is blockchain. And in fact, I have projects to use blockchain for data integrity and data and for audit purposes and so on. Because you see, with the extent now of testing, you want to make sure that the data you collect, maybe your IRT test and so on, are really the data are correct and you know where they come from. And especially when you're going to go in the region, in South region, you want to make sure that the data collected in Myanmar and are good. They cannot be falsified. And then you could go back to the owner of the data and even go into deeper. And so blockchain is going to be one of the possibility to really have data integrity and for audit purpose when you want to do this massive surveillance of data, not massive surveillance of people, massive surveillance of data.
Yeah, that's interesting. Yeah. So one additional note is that I know that a lot of finance people use the portfolio theory, try to minimize the risks for the medicine industries to diversify the risks in medicine development or in vaccination development. So that could be also one collaboration across different disciplines. Okay, so let me pick up a question from the audience and switch a gear a little bit. So there's one question from the audience. It's like, will boosters be required for an indefinite duration? And what is the impact to human body from too many booster rounds?
That's a smart question. Booster maybe will be needed if the immunity wanes progressively. The problem, I don't think realistically that it is possible to implement boosters every 3 months or every 6 months or I don't think logistically it's going to be a nightmare because you're going to have to produce billions. And I don't think this really, will be efficacious. Then we will need really to think of what I was calling the vaccination 2.0, new type of vaccine, because this is not a sustainable strategy to have repeated booster. I mean, number 4, number 5, and so on. I don't think this will be a viable approach.
Thank you.
Thanks. There's a very interesting question about different policies adopted by different countries against COVID-19. So I think there are maybe at least 2 approaches to dealing with the pandemic. We have the zero COVID approach, say by China, and also the allowed to become endemic approach adopted by many other countries at the moment. So which approach make more sense to you based on the scientific evidence?
Make more sense now to me now, but at the beginning, nobody could say he has the right approach. I mean, if somebody had the right approach at the beginning, we will not have 350 million cases around the world. So now what makes sense for me is that we're gonna have to live with the virus. It's impossible to control, because the moment you reopen, it will find a way to go in. So again, it's like for boosters. Are you going to do boost of closing, opening, boost of closing, opening? I don't think this is either politically or economically viable. So I favor the first one, which is progressively relaxing and learning to live with the virus. Especially the main question for that is this virus is going to be less severe, but it will be contaminated, but less severe. If it's the case, it will be easier to live with it. And if it's still severe, but we know which population are at risk, focus on the population at risk. And we live with it.
Okay, that's a very interesting. So a related question is that one people ask that the SARS, 2003 SARS, disappeared by itself, and why the current COVID-19 situation is hanging around for so long, and more importantly, does Omicron suggest it will get into something more like that we can get back to the normal soon?
Okay, so viruses don't disappear by themselves. If it was the case, it would be very good. Could wipe them too. No, but the reality is that that's where knowledge on the biology of the viruses are important. When SARS happened, it was realized very fast that the moment people were infected, this moment they developed the sign of infection very fast in the 24 hours post-exposure. So if you have a surveillance system where you say, okay, these people, they are the signs, they come to the hospital, you know they are infectious, you isolate them, because for SARS, at the moment they were infectious, they were infected, they were infectious and able to contaminate people. So if you have a system where you could identify them very fast, isolate them, you prevent transmission. SARS-CoV-2 is a more tricky virus. You could be infected for 1 to 14 days and being completely asymptomatic, which means no fever, no sign, but you are still transmitting. So unless you do like in China, you test millions of people and then you isolate them. But you see, again, what you need as a system to do that, then you could prevent the transmission. But still, during 1 to 14 days, you could transmit the virus. And SARS, first day, we knew you had the signs. With SARS, you always have the signs. Very, very rare asymptomatic cases. So that's the biological difference. And Omicron, yes, luckily for the moment, there are more transmission, but less severe cases. And so if you consider that this few number of severe cases, we are back in a flu season. When, flu season, high volume of transmission, everybody has running nose, not feeling very well, but you stay home for 2-3 days, or sometimes you even come back to work. You contaminate your colleague, but you know that you're not going to have people dying too much of that.
Okay, then I have a question kind of related to my role in the school, also related to your role in the university. So you mentioned that this pandemic actually revealed the weakness of scientific education. You believe, I think in one of your slides, you mentioned people need to have more knowledge about public health and social and economical impact, etc. So what do you think the implication of this pandemic on our education sector in general, and also the business education in particular?
You know, for example, if you have in secondary school and having 1-2 hours of what is a vaccine, how does it work? Not going into the nitty-gritty details of the immune system, but what is a vaccine and so on? What was the situation before vaccination, after vaccination, and what is the side effect? Okay, and explain really the concept of risk benefits, maybe not in secondary school, but after. Then people have a basic knowledge, so they could make educated guess and decision. That's, I've never been for a vaccine mandate in my life, except for the vaccine for the kids, because we know for the vaccine for the kids that if you don't vaccinate smallpox for the kids, the mortality rate is 40 to 60%. So this is very high toll. But when you have diseases where the case fatality ratio is very low, I more than convince people, educate them, convince them. And that's where you really have to fight with all the crap that you find in social network. And if people are educated, you know, they will take also some distance with the hearsay, all the craziness that you could find around here.
Yeah, the scientific knowledge indeed is very helpful for us to make informed decision, even for business
decision is important.
Yeah.
Yeah, okay, so next question. I just saw this question from the floor. It's related to the previous one. So what do you think of having children under 12 years old vaccinated with mRNA? Obviously, a lot of people are thinking about this situation right now and it's ongoing. So yeah, it will be good to get the suggestions from you.
I guess you're also asking it on your behalf because you have children younger than 12 years old.
No, they're all,
yeah, that's right.
They're old. The last one is 20, so he's 20 and he's vaccinated. So but he decided by his own, he didn't listen to me, so it's okay. This is a tricky question. And again, it's really deciding on risk benefit. And I can tell you, we had a lot of discussion of the different committee reviewing all the evidence because we didn't want to make a hasty decision for that. You know, the implementation of vaccine was really in June last year when Singapore bought the vaccine in June, and it started to implement in January 2021. Okay, and we waited the longest possible for data coming from the different clinical trial in the US and Europe before deciding to go for vaccination of the kids. And you could see also we did it in stages. It was beginning 18, 55, we went to 16, then we went to 11 and so on, then before moving. It's based on data and really appreciating the risk benefit. At first, and to be honest, and again, I want to make a disclaimer, this is my opinion. It doesn't cover the opinion of the different committee and the government. At first, I was not so much inclined of the vaccination for below 16. I didn't see the risk benefits and so on. But progressively, there have been cases on kids, you know, and then you have to say, okay, it may be these numbers of cases might be limited, but if there is a parent where, because you have not allowed them to have the vaccination, and he comes to the hospital and die of COVID, what will be the population response to that? That's one aspect. So there were these things, we need to protect kids, and there are, and in particular in COVID, there is a disease called MIS-C. I will not go into details, which happen in a particular subpopulation of kids, and which could be very, very bad. And we had reports of, in the US, in Europe, of newborns dying of COVID, of baby dying of COVID, of overweight kids in the US below 16 died of COVID. So there were really a concern of that. Then the other point, I'm sorry, I'm a bit long because I want to make you really understand that it was not a decision like, oh, we're going to extend to vaccinate everyone. It's really, I've been really thought through. Then the other part is side effects, because really the thing you don't want is that you vaccinate a kid, is in good health, and he has deep side effects. And we know these vaccines give side effects. You know, I've been receiving my boost last Thursday. I was out for 2 days with pain, with fever, and so on. You don't want to put your kids through that. That definitely. So again, we look at the number, and when you look at the number, and that the thing, I know it's the most difficult thing, and I have a lot of discussion with friends about that. When you look at the population, the side effects are very minimal. I say at the population. The benefit will be better for vaccination with kids at the population versus the side effects. But if you are the parent which your kids receive the vaccine and have side effects, you don't care about the population. The 0.01% of the population for you is 100% is your kid. So that was a lot of discussion. And at the end of the day, really the consensus that we need to vaccinate kids for this. And also because especially with Omicron, you see the virus are smart. At the first of the epidemic in 2020, kids were not very infected. If you look at the number, you don't have that many kids infected. And progressively with Omicron, you see the number of kids infected growing and growing. So the virus is adapting to the population also.
Yeah.
The idea of the
yeah,
that too.
Maybe we have one more question before you wrap it up.
Sure.
Yeah, because there's a question from a final year student at NIE. The question is about this vaccine differentiated measure in Singapore. So given that these measures limit people's freedom from socializing and frequenting public spaces based on one's vaccination status. So what's your view on these measures?
Again,
social perspective.
Yeah, it's a public health measure. Do you want to have? There is one moment you need to control dissemination. Okay. Again, I know people take a snapshot, but at the beginning, when the vaccine, we had data for the first wave of virus, and we know that the vaccine was really limiting dissemination. So that's why it was important to differentiate people with vaccination, not vaccination. And I'm not a vaccine crazy because my wife cannot be vaccinated because she has allergy problem. So the doctor would say you don't go for vaccination. So I know what is the life of not being vaccinated. She cannot go in many places, so I don't go because she cannot go. So I know what is that, but you need to prevent dissemination. And you know that the non-vaccinated are the easiest pool of people carrying the virus around. And it was true again with all the previous variants. With Omicron, the things are changed because Omicron doesn't care you are vaccinated or not vaccinated, it disseminates. So I would say 6 months ago, it was important to implement. With Omicron, it's not that much important to implement. Again, it's my point of view.
Okay, thank you very much.
Yeah.
So I think we're right on time, and I just want to thank all the participants today for such a very insightful, engaging discussion during the Q&A. And especially I want to thank Professor Renia again and also our NBS Knowledge Lab who organized this event. So this webinar is part of our NBS Knowledge Lab series of webinars, and we hold this webinar every month. And to look out for upcoming, so please do to look out for the upcoming webinars and stay connected with us via our social media channels. And last but not the least, do take a few minutes to do a quick survey at the end of this webinar. Thank you very much.
Thank you, Lauren. Thank you.
Thank you.
Mhm.